Well, thank you. I'm happy to be here. I really appreciate the opportunity to talk to you. And, you know, we're going to talk a little bit about our SU canola. That sort of comes toward the end, because what I wanted to do, I think, is very important to understand the technology. Technology is developing and changing. You probably hear a lot about it. Are there any molecular biologists in the crowd? Good, because otherwise they'll call me out on it. But there's some pretty cool stuff happening here, and I want to talk about that and how it's happening, and then what kind of products it's leading to. So what I'd like to do is I'll give you a little introduction to our company, talk about the technology, and how all these things fit into what we'll eventually call and talk about as gene editing. Then talk about Cibus innovation. We are innovating using these new technologies, developing traits in crops, and then talk a little bit about the future. So first of all, let's talk about Cibus. Cibus is a trait development company. We're headquartered in San Diego. We've got offices in Minneapolis, where I'm officed, Capella in the Netherlands because of the technology more than anything else, and most recently, Winnipeg. So we're expanding our footprint all along, and we are continuing to look for opportunities throughout the world. We're focused on improvement of plants. Improvement of plants through precision gene editing. And you know, one of the other operating units we have is called Nucellus, and they use the same technology to work on yeasts and things like this. So any organism that has a gene, you can use this technology on. And so people are even talking about using some of the gene editing techniques on people, on animals, to cure diseases and things like that. So it's pretty impressive. I won't be talking about that. I'll be focusing on plants, obviously, because that's our expertise here. Well, you know, why are we doing this? And I think you heard in the opening presentation today, there's a population that continues to grow, and they need to be fed. So how do we do this? And how do we do this giving them what they want? Okay? A lot of times we say, well, hey, we can grow this or grow that and provide this, but the consuming public needs to be able to say, hey, yeah, I want this or I want that. That's where we're facing to some extent in the case of our demand for non-GM food crops. So we're looking at meeting this demand. We hope we can use our technology along with other things to help do that. And Cibus really wants to be a leader in the development of sustainable, global, non-transgenic food supply. So let's talk about, you know, how you get there. First of all, we have to understand, I mean, there's a lot of diversity out there. As a plant breeder, I've been breeding plants, breeding corn primarily, most recently canola, for over 30 years. I've seen diversity, and you can see it in the corn here, tomatoes, the carrots are amazing. I had a few of those in my garden this last summer, and you know, you can see a whole bunch of diversity. Of course, in the animal world, you see the same thing, right? So that diversity is what plant breeders in the plant side have worked with to develop the varieties that we have today. That's very, very important. In fact, if we look at a simple example, look back and say, okay, what happened in the simple area that's related to canola? In the Brassica species, from a simple Brassica oleracea progenitor, we see that there's a significant amount of variation. I'd use this thing, but I can't see it myself. Oh, there it is. Okay. I'm colorblind, so I don't see the reds all the time. But anyway, you can see some very significant diversity in here that just came from one crop. And how did it get there? Well, it's driven by mutations, okay? Changes in the DNA that have happened and accumulated over years. And so this is an example that we had. This is from a paper published in Science, and it was regarding Arabidopsis. Now, Arabidopsis is actually a little weedy-like plant. The scientists like it because it grows very quickly and very quick generation turnaround. And so what they noticed is that if you just propagated the plant the seed, let it grow, harvest the seed, plant it again, and keep on doing this, they did it for 30 generations. They looked closely using molecular tools, and they found 116 different changes. Okay, that's significant. And that's just from growing seed, taking it from generation to generation. So this stuff happens. It's natural, naturally occurs in the development of plants. And, you know, we've seen this in the plants that we might be familiar with, things that we consume. Grapefruit. That's not a particularly appealing grapefruit. I hate those when you get all the seeds in there. But we've had seedless grapefruit develop over the years. In the different crop species, a lot of variety. How many of you remember growing wheat that grew 6 feet or so tall? Anybody in here? Nobody. Okay, there was wheat around like that. It did grow that tall. Well, it was that tall at some time of the year. Around harvest time, it was usually about this tall or maybe lower because it lodged pretty badly. And semi-dwarf wheat came around and really saved the industry there. And so it's been a very important development, these mutations, these small changes that people have found and taken advantage of. Now, breeders have realized this. They understand how valuable genetic diversity is, and they said, well, okay, what about tools? Can we have tools? I mean, you can just plant stuff out in a field and you can look for mutations. I used to do that in my corn nursery. I planted thousands and thousands of rows, and every once in a while you found a plant that looked different. And you typically said, hey, I'm interested in that one. I'm going to keep that one and see if it can add some value. And so you do that. Well, there's other tools that breeders have used over the years to be able to actually create these changes. You can add certain chemicals to the pollen and then pollinate using the chemically treated pollen, and you can see more of these changes happen. You can use radiation. Radiation actually makes changes in the DNA also. Well, okay, you can see the circle one. That's the mutation I want. That's something I'm interested in as a plant breeder. But when you do it this way, you have a whole lot of collateral damage. Have a lot of other mutations that occur there. Some of them are really problematic, and you got to get rid of them. Other ones are just sort of hanging in there. They do affect the performance of your plant. So they're really not positive. They're not things you want to have hanging around. And so what you really need to have is something which allows you to say, okay, I want to make that change and only that change to change the trait. And so it is very important to be able to say, okay, I want to make a single change. Well, but then if you think about it as a plant breeder, I know that there are very few traits anymore that are just controlled by one gene. There are multiple genes around. So you want to be able to say, okay, well, what about other areas? Can I make multiple changes in that genome and not have all those other changes that we saw on the previous slide? And so the key thing is to be able to make these changes and then find some value. And you know, when you change the spelling of genes by modifying one or more of those bases, you can lead to a number of different things. And we've seen herbicide tolerance, oil protein quality, even disease, insect tolerance, starch quality, heat and drought tolerance, many other traits. You name them, they are obviously being conditioned by genes. And so if we find out what those genes are, then we can change them, and we can change them in a way that gives us value, add some value for the program. So the whole thing about gene editing, you know, we really needed a tool to say, okay, we don't want to make up all these changes and have all this collateral damage. We want to be able to have focused changes with no off-target effects that can allow us to make those small changes that lead to large effects. So enter gene editing. That's the technology that happened. And, you know, sometimes it's hard to understand if, you know, a small change, I mean, one letter change, can it actually affect much in the genome, in the plant? Well, let me give you this example. Okay, I like bears. Bears, they're cute but scary, carnivore, right? I mean, they're a problem. But if I want to edit a word there, I can change the entire meaning to a refreshing adult beverage. Okay, so in one case, you know, you'd be, you know, probably ripped apart, and the other one you get, well, little crazy. But anyway, you may have a very, very significant difference there between the two. That was only a one-letter change. And in the same way, gene editing can make small changes in the DNA that can have large effects on the organism. There are a number of trait development technologies. As we look back in time, plant breeding, of course, has been the primary one, and it continues to be the core of the development. Now, there are other tools that plant breeders have developed and used over the years to allow them to become more efficient. One of those tools is making transgenes. And of course, a transgene is when you take a piece of DNA from another organism, you cut it out of that organism and put it in the one that you're interested in. Okay? Wasn't there, it never was there before, but it's there now. And it has some value, right? I mean, we've seen some herbicide tolerance, we've seen some insect tolerance that certainly were part of that transgene technology. It creates some uncertainty, though, in the sense that you cut that DNA out, you throw it in there, and you say, I don't know where it lands. And you hope it lands in a good place. And so technologies have been developed that they try to fix that, but it's still a pretty imprecise science. We talk about, and I'll talk about the top one because that's where the Cibus technology is in another couple slides, but that's where you have actual editing happening. Some of the things that you've all often heard about too are talons and CRISPRs in particular. You might have read about those being the new wave of editing. And I'll talk more about that later, but these are the tools in the toolbox right now. These are what we have available to us that allow us to make traits and improve plant species. Now, talking more specifically about the Cibus technology, really the technologies developed over time have come together to allow us to be much more effective in developing our technology. Realistically, it started with genomics. And what is genomics? Genomics is very simply the study of piece of DNA, what that piece of DNA does in the plant, okay? What product does it make? Does it make an enzyme, a protein, a fatty acid? What is it responsible for? Okay, people understand that more today than they ever have before. And what that has allowed us to do is it allowed us to understand what genes are important, and if we want to change those genes, we know what they are, okay? And so that information, well, then you have tools, and I'll talk more specifically about the Cibus tool, that you can use to make gene editing so you can actually change those genes and make the changes that you want. The other very important part here is cell culture. And the reason that's important is because if you have a whole plant there, it's really hard to make a change of everything in the plant that you really need to have changed. And so you have to reduce it to a fairly simple state, and that is at the cell level. We actually take our plants that we're working with, and we reduce them to a single cell. We make the change in that cell, and then, of course, we have to bring that cell back into a full plant. And that's part of the whole technology package. What we do is we make traits in plants. And here's an example. You take a red flower into a white flower, and you say, okay, I want to make the change. I'm going to use this technology in there. We reduce it. We take it from the leaf down to a cell of that leaf, into that cell, to the nucleus, find that gene we're interested in, and we target that gene. More specifically, what we do is we develop what we call a gene repair oligonucleotide. Yes, there will be a spelling test afterwards. Gene repair oligonucleotide, it's a GRON. We call it a GRON, so we simplify it. But what that is, it's synthesized in the lab, and it's synthesized to identify that area of DNA that has the trait we want to change. And it's synthesized in a way that we build the change into the GRON. Now, you notice we also have a couple things on the ends of the GRON. Well, that's a very, very important, because if they're not there, the DNA could incorporate it in becoming a transgene. We don't want that. We simply want to edit the gene and make a new trait there using natural cell capabilities. So what happens? We introduce this GRON into the cell. That GRON actually finds the DNA. Now, how it does that, I don't know. I'm not a molecular biologist. But there are the ability for that DNA to actually find its counterpart. It finds it, it pairs. Now, when it pairs with it, it makes what they call a mismatch bubble. Okay? It obviously, that what we want to change is not in the DNA to begin with, and so it's a mismatch. Now, there are enzymes in the plant that are there to actually find these mismatch bubbles and to correct them. Why are they there? Because they're protecting the plant from mutation. Otherwise, the plant could have mutations. So we talked about that well earlier, that plants it just happens as part of DNA copy or external factors, you have mutations show up. So you have these enzymes in there that protect the plant against mutations. And so what that means is that it finds this mismatch bubble and says, oh, hey, there's a problem here, I'm going to fix it. And so they basically go here, identify it, fix it, put the new base in, and then you have a heritable trait. The GRON that we inserted only has a fairly short half-life. And so it lives in the cell 45 minutes to an hour or so. That's basically all it's there for, and then it's degraded by the cell. Now, why is that? Because the cell doesn't like extraneous DNA hanging around there. It says, well, that might be a virus, and I don't like viruses. It creates problems for me. So you have all these factors, but we're taking advantage of this system that's already in the cell. So it's a natural system that's there to make the changes. So when you use this technology, what are the advantages? Well, first of all, it's non-transgenic. There's no DNA that's added that remains in the cell. It's very precise and very specific. It goes right to the gene that you want and makes just the change there. And I'll show you a little bit about that because it's important to understand that. Only the targeted base, as I mentioned, it works with the cell's natural systems, and you can successfully apply it to a number of organisms. Now, and we've had some experience with multiple crops that we're working with at Cibus. Any crop really is possible, and the key is develop all the system around it. So the cell biology, the molecular biology, and all the pieces to be able to take that through to a final cell. But as I mentioned also, yeast and other microorganisms, you can use those and change and make traits in those too in the same way. Now, let's talk about some of the other tools that I'll talk about very briefly. They're pretty cool. I mean, there's some amazing science here that allowed people to identify these various nucleases is what they call. They basically typically have a string of DNA or RNA that help them find the target, so they go right to the same right place, and then they do something to that. Well, typically, all these things, with all the cool pictures and everything else that you see here, they do one thing. They cut. So they find a piece of DNA and they cut the DNA. Okay, so what does that do? I mean, is that editing? Yeah, I guess it is, but it's sort of a random edit. So what happens is when you cut it, there's two things that you could see happen here. One of the things is a gene disruption or a knockout. So in other words, the gene that was there that used to be functioning is not functioning anymore. Okay? And there's a number of traits that if you knock out a gene, it actually has a beneficial effect. One of our competitors, Pioneer DuPont, is working in corn, and they're finding that there's a gene they knock out to make waxy corn. It's a certain type of corn that has different characteristics, and they're able to do that very efficiently with this technology. The other thing that you can do is you can actually add transgenes. So you can add DNA and make a really targeted transgenic. So it solves the problem that the transgenic guys have had to say, okay, I don't know where my gene goes. I can pick a region in the genome that's consistent, that's very stable, and I can put the gene there. But the challenge is you can erase words or genes. You can insert new words or genes, but you cannot make the single letter changes. So you need more to make those precise changes. And this is where the Cibus technology comes in. So not only does the technology work by itself with the making changes, but we also can work with the double-stranded breakers, with the cutters, and then we add our GRON. That tells the cut how to heal, okay? It's sort of like a Band-Aid. You put the Band-Aid on there, the Band-Aid allows the cut to heal, right? You don't do anything else, you don't add anything to that. It simply is there to help affect the change. Non-transgenic, because the materials don't stay there, and it is a precise, single and multiple letters can be intentionally changed. The gene keeps operating. So we don't knock it out, it keeps operating, the right place, the right time, and gives us the trait we're interested in. So just to review the technology portion, in RTDS, we can just use our GRON only, and it can give us different traits. Also with the cutter, we can still get the traits, you get the same result. They're precise edited changes. In fact, if you think about this, and we like to think we're inspired by nature, because we believe that if you look at it, we direct the change. Oh, nature directs the change. Okay, that's fine. You use the same natural DNA repair process. In nature, it'll probably be millions of plants that you have to sort through to figure out where the trait is. We look in cells. And then it's very fast and precise, long time with nature. So we can get there. And the interesting thing is you get to the same place. The traits that we develop versus the traits that would come naturally through just growing the plants and searching through them, same thing. Now, a key question then becomes, okay, how's this regulated? Is it regulated? Are there issues throughout the world that we have to be concerned about? And there certainly are, of course. As we develop any new technology, people come up and say, well, wait a second, I'm not sure, can you grow this? We don't understand it. So we've been spending a lot of time throughout the world working with regulatory agencies and bodies to help us, to help them understand it, and then to approve the process. And so we're making progress throughout the world on this. Particularly the most challenging one, of course, is Europe. And the European Union is, well, I think it's that close. I would have said that 4 years ago too, but it's that close to coming up with a ruling that would be favorable toward this technology being used throughout Europe. They were supposed to have an announcement on the 20th of December, but in typical European fashion, they pushed it back until the 18th of January now. So we're still hoping, but we're waiting for that final approval. But other areas, in fact, there are 6 countries in Europe that are already said to us, hey, if you want to grow your stuff here, it's fine. Because we believe this is an alternative way to create mutations in plants. It's just plant breeding, but it's more precise plant breeding. And we like that. So it's all good on paper, but let's talk more specifically about, okay, how do you get it done in a plant? And so we published a paper a couple years ago about the work we were doing in Flax. And so I can talk a little bit more about that. I'll reference some other things, but I'll also talk a little bit more about our project here. The Flax Council of Canada came to us and said, we got a problem. We've got a contamination problem. We don't want to have that, but we still want traits in the crop. So how do we go about doing that? So we said, sure, we can take care of that for you. We'll work on it. We developed the system. And so all the way from single cells to regenerated plants in flax, we were able to generate that system. We made the changes in the flax plants, and then we said, okay, this is a herbicide tolerance trait that we were looking at, by the way. We said, let's try to put the herbicide on, and lo and behold, the plants that we had created that had the changes, our targeted changes that I just sort of talked about that technology, were fine. And yet the control crisped up and died. We looked at that a little bit closer then, and from the molecular biologists, they love to look at all the spelling, essentially, of the genes, and they took a look at this, and you can see that here's the before, there's 2 C's there. Well, we changed to a G and a T. So we made the changes, and those were the changes we wanted to make, And that worked. Now, there are 2 genes in flax that condition herbicide tolerance. And unfortunately, the second one in this case was not changed. We looked at that, and that was fine. We still advanced the product to take a look at it. But then we also asked the question, okay, we don't want to see any collateral damage. We don't want to see other mutations happening. And so we looked throughout the genome, particularly at some very important areas that match. Now, the molecular biologists are telling me, oh yeah, that's what we do, and I believe them. So you just have to trust me on that one, because that's the best I can do for you. But the bottom line is they found no other mutations. So they didn't find any other changes. We were able to use the technology, just make the precise changes that we wanted, and that's all. So bottom line is we don't have that herbicide-tolerant flax out. It's glyphosate tolerance, but that single gene change wasn't quite enough. It made sort of a cool little thing. I call it ornamental flax. They look like little Christmas trees in the field. They never quite grew out of it, though, but they didn't die. Okay, that was sort of interesting, but obviously we're not going to make money on that. So we went through back to the lab. We said we need to make that second gene change. We've got plants that have the second gene changes, and we are just finishing those up in the greenhouse right now. So we expect and hope to be able to have some flax plants in the field next summer to be able to confirm and say that we do indeed have the glyphosate tolerance that we were targeting. Canola is really the best system we have in place right now. We've been working on it the longest, and so from a protoplast all the way through shoots, it goes very, very fast. In fact, the last traits that we've been working with in canola, it took us about 6 months to be able to take it down to a cell back to shoots. That's not that long. So it really allows us to move quickly, and right now, in the top 3 categories anyway, other herbicides, oil modification, yield retention, we've got plants that we're looking at, and we're trying to see how they do and how they perform. And so disease resistance is another area we're working in. We're looking at sclerotinia, blackleg, clubroot, among other things, to see whether we can find some genes and pathways that we can change to affect that. And canola continues to be the leader in this area. We're also working in grasses. So rice is one that we've been looking at. There's 2 herbicide tolerance traits that we're working with in rice. Now, you're not going to be growing these in Canada, at least for the next 5 years until the climate change takes over. Then you probably have some rice paddies down somewhere. But anyway, we're growing rice. We've got some plants that show the trait, and we're just to the point of trying to get some seed from those plants and testing those further. So that's moving through the system quite quickly. Potato is an area that we've worked in, and potato we started with there, focusing primarily on disease resistance, and Phytophthora in particular. And this has been quite exciting, because what we've learned in potato, we can actually apply those learnings to working in canola, flax, and other crops. So this is a pretty exciting one, and we've got some regenerated plants coming out of that system also. As we've been developing and working on these materials over the years, we've been more efficient. You know, when I started 10 years ago, it was sort of hit and miss. I mean, we were a little bit better than nature, but not much. Well, right now, we've gotten to the point from 2014 up to 2017, you can see a very significant increase in the number of conversions or mutations that we're actually seeing in the plants. The people in our lab keep on threatening me. They keep saying, well, you're in charge of product development, which means that when it becomes a plant, it becomes my problem. They said, we're going to bury you in plants. Well, they're getting close. And so fortunately, I've been able to hire a few more people to help me with that. But we're getting to the point that we're getting a lot more plants coming through the system, which is quite exciting for Cibus. You know, and the one way we look at this is we say, well, this is sort of like a trait machine. We're going to find a variety, the best variety we can. We put it at the front end of this, and now we're going to hit the program, and we're going to say, well, okay, I'd like some higher yield. I'd like this type of herbicide tolerance. Oh, this disease resistance. And this quality characteristic, okay, push the button and boom, we got plants coming out. Okay, we'd like to be there. We're not there just yet, but it's close in canola. It's close in canola because we're able to take some of the most elite materials, put them in at the front end of the pipeline, and take them all the way through. And as you think about as we're developing this trait machine, there are a number of things that have to come together still, and all the pieces and parts and everything like that. I mean, the first and foremost goes back to that early slide that we talked about genomics, understanding what genes are there, what have to be edited to make the traits we want. Then secondly, well, okay, we know what has to be edited. What change do we have to make? What letters do I need to change there to actually make that gene exactly as it needs to be and to deliver the trait that we want? And then, of course, that plant cell culture thing. You know, that's a lot of work. Rice is behind canola because rice is very difficult to work with in the laboratory. But we're getting a whole lot better at it, and we have had this experience with multiple plants. And so we believe that that, along with other grasses, maybe a wheat or a maize at some point in time, should be able to move very quickly through this program. From Cibus perspective, we've got some products coming through right now. Our SU canola is certainly a first product, and you can learn more about the SU canola at our booth downstairs, or no, this level, wherever it is, at the booth in the main area, but you can learn more about it. We've got a hybrid that's registered right now in Canada, and we think there's some exciting opportunities there. The one interesting thing about the non-transgenic nature of it is that Cargill, Bunge, both we've been working with, and they both are interested in growing that segment because they are getting a significant amount of demand from the consumers. And so this creates a real opportunity for us to be able to service that market. Flax, we expect to be happening in a couple of years probably, but it'll be the first non-transgenic glyphosate-tolerant crop on the marketplace. Rice, again, I mentioned the herbicide tolerance, and then Phytophthora and potato, all of which are in our developmental pipeline at Cibus. SU canola system, I'm not going to talk a whole lot about it, but one of the things that we've been happy with is that it's economical, it's easy to use. People understand SU herbicides, there's a nice fit in the rotation if you have Roundup Ready soy. Roundup Ready soy, Roundup Ready canola, they don't work together too well. And so SU provides another alternative to Liberty. Liberty is the other option. It does work in that kind of a system. And as I said, that demand keeps growing. I can't say why, but the thing is that as producers, as developers, we have to ask the question, well, okay, if somebody wants it and we can provide it, why not? If they're willing to pay for it, let's give it to them. And so we're not going to necessarily argue the benefits or the non-benefits of transgenic versus non-transgenic. Hey, we have the tools, we can deliver on those things. And that's one of the things that excites us at Cibus. We can address not only the North American, but also the global market. Outside the US, there's even more skepticism, let's say, of GM products and things like that. We got a technology that will be able to allow us to go beyond that. And we're moving quickly. I talked about the innovation of plant breeding tools, which is our editing capability. Along with that, you got to be able to protect your intellectual property. So we have probably 350 issued patents to protect parts of our technology. We're looking at any type of markets, whether they be new markets or existing markets. I mentioned the piece about regulatory. We're getting some positive responses in different parts of the world, so that's very favorable. And the product pipeline is robust. We're getting a number of things coming through it fairly quickly. So in final summary, we intend to continue to develop crops, mutation-based traits, in not just those few crops that I mentioned to you, but we're also looking at moving out into other crops that are important to Canada and other parts of the world. And the sky's the limit. As we learn more and more about the genes that are effective in environmental stress, yield, some of these quality, HT, all of that piece, as we learn more and more, we've got the tools to be able to address it. And we can make the changes that people will want to see in improved crops as we go forward. We hope we have the ability to do that, or the capability and the opportunity to help feed the world, because that is still the main driver for us. Thank you so much for your attention, and I would ask if there are any questions.